Oh, thank you very much for the invitation, Nima and the organizers. It's a great pleasure being here. So I will be talking to you about a number of technologies that our lab has developed over the last 10 years or so, in part funded by the BRAIN Initiative. But sort of the broader questions that our lab, and I guess many other labs are interested in, are the following. So how are sensory inputs represented across the brain? How does the large-scale activity of neuronal population is generating behavior, and what is ultimately some of the underlying computational principles that the brain uses in order to represent and process information? So if we consider these questions, we can ask also a simpler question, and that is, how does the scaling of information looks like in the brain? So how is the information distributed basically across the population of neurons within the brain? So to make it more concrete, we can look at this representation in which we are looking at the amount of information that the brain is encoding versus the number of neurons that I am looking at. And so one possibility that we can think of is that there is simply an unbounded scaling of information. As we add more and more neurons to the population, we simply encode more information. But there are also good reasons to believe that this scaling may not hold arbitrarily, and as we add more neurons, that we at some point really face a bounded scaling law, and there is some evidence for that in the literature. The problem that we are facing, however, with, say, commercial neurotechnologies that allow us to record maybe from something like 100 neurons or so, it's very difficult to know on which of these scaling laws we are moving forward. So that is one of the motivations that we want to push the limits on the scale at which we can record from neuronal population, because this answer is really important, because if you imagine these are a group of sensory neurons, the type of information that these neurons are receiving and the way the information is represented will determine some of the downstream availability of information for other brain regions. Another reason is the fact that neurons are densely interconnected, and these interconnected neurons can span many centimeters or millimeters, depending on the model systems, across the brain. And so to make that a bit more concrete, so imagine that these would be the responses of 3 sensory tuned neurons that happen to be in regions of the brain that are not accessible at the same time. So you are forced to record from them in a sequential manner, and if you do that to these different representations of the stimuli, you may be led to believe that they are doing the same thing. However, if you were able to record from them all at the same time across trials, you would see that they exhibit a lot of trial-to-trial variability. And not only that, but also that some of these variabilities are correlated. So there is covariation between neurons to the representation of the same stimuli, which are referred to as noise correlations. And we know this is not really noise, but it is capturing information in the brain and how sensory inputs are interacting, for instance, with internal states to drive behavior. So this is another motivation for why we want to be able to record at larger scales and from many neurons as possible. And I hope to give you a little bit of a sense of what it takes to scale up neurotechnologies to larger neuronal populations, and the kind of insights that we can hope to gain from the brain. All right, so this has been the focus of the lab really for the last 10 years or so, where we initially started developing these technologies in the smaller, more tractable model systems like C. elegans and larva zebrafish, where whole brain recording seemed to be within reach. And then over the last 5-6 years, we have pushed this more in the more scattering mammalian brain, mainly the mouse brain. All right, so since many of you are probably familiar with the standard tool of recording neuroactivity in the scattering brain, which is two-photon microscopy, at least when it comes to optics, let's talk about what are some of the fundamental limitations of diffraction-limited two-photon scanning microscopy. So why can't it be scaled up to record from many thousands and whole brain from the whole brain? So let's imagine this is the size of the volume that we want to record from, about 500 micrometer cubed, and we want to record from this volume at something like 5 hertz. And if this is the size of our diffraction-limited voxel, So simple math shows us that that translates to 500 million voxels, sampled at 5 hertz. And since each voxel needs to be excited at least by one laser pulse, we need to have an acquisition rate of 2.5 gigahertz. And and that means that we need to have a laser that runs at 2.5 gigahertz repetition rate. Now, such a laser is not very conventionally available, but even if it would be, there is a bigger problem. In this scheme, we end up dwelling on each of these voxels for only 0.4 nanosecond, which is an order of magnitude shorter than the fluorescence lifetime. So we are not able to collect any fluorescence light from each of these voxels. So it's not a matter just, as you can see here, in speeding up acquisition. So one question that we asked a few years ago is, can we relax the spatial resolution without losing information? So if you look at the mouse cortical neurons, which are about where the somata is about maybe 10 to 15 micron in size, and compare it to the size of the diffraction-limited spot, you may see that this is maybe unnecessarily high of a resolution. So maybe we can relax it and still be sampling these neurons within Nyquist limit. So for instance, by underfilling the microscope objective, we can create a spot that is about 5 micron in diameter, which would be still faithfully sampling these objects that are 2 or 3 times larger. However, due to limitations from optics, you will end up with an axial localization of the excitation that is on the order of 70 microns or so. So you would lose all sectioning capability of the 2-photon microscopy, and you will not be able to discriminate between neurons that are on top of each other. So this led us to the idea of light sculpting, where we came up with an approach, for instance, by using the technique of temporal focusing, in which one could use a temporal degree of freedom to sculpt basically the distribution of light in a more arbitrary fashion in the sample. And now, if you have a spot that is now about 5 micron wide and 15 microns long, and you scan this now simply in X, Y, and Z, and go back to the same example that I started with, the same volume is now translating to 1 million voxels, and sampling that at 5 hertz means that we need just to have a laser that runs at 5 megahertz, which is much more conventionally available. And more importantly, we are now dwelling on each voxel for about 200 nanosecond, which is sufficient to collect all the fluorescence light out. All right, so this was we developed this technique a while ago, and then we asked, okay, what other things can we do in order to further speed up acquisition of voxels within the sample? Now, if you visualize now this situation with the 5 megahertz repetition rate laser that I talked about, you will see that these are the individual pulses, so they are separated by 200 nanoseconds. And after the excitation of the sample with one pulse, about 5 or 8 nanoseconds after that, the entire fluorescence light is emitted. So if you look at this schematics, you're seeing that for the majority of time, there is no exchange of photons between the sample and the microscope, so we are just waiting for the next pulse to arrive. So we can ask, wouldn't it be more efficient if the next pulse would be coming right after the fluorescence has decayed, and then the subsequent pulse, and so on. So this is the idea of temporal multiplexing. So what we can do is we can start off with a much stronger pulse, then split it by using a series of beam splitters, and then let each of the sub-pulses propagate over a distance that corresponds to a multiple of the fluorescence lifetime. So these pulses would be each separated by each other by about 8 nanoseconds or so. Now we can bring these pulses down now together and focus them either onto different axial positions within the sample or to different lateral positions. And then when we scan them all at the same time, we can just look at the arrival time of the photons, and then we would know which plane they would be coming from. So this speeds up acquisition by about 4-fold, because we are having here now a 4-fold multiplexing. And this is an example of that. So here you see 4-fold lateral multiplexing, in the mouse auditory cortex, covering a volume of 1 millimeter by 1 millimeter by 600 microns, containing about 12,000 neurons that we can now capture all at the same time and record. I believe this is this is at 5 hertz. All right. So what are some of these design concepts of that are needed to do large-scale or scalable volumetric calcium imaging? So one of them I mentioned already in passing, and this is we want to excite each sample voxel exactly by 1 pulse and not more. And the reason for that is that for the same amount of average power, using a 1 pulse per pixel excitation, you are creating the maximum amount of fluorescence light, right? Because if you have multiple pulses, then the signals will add up just linearly, whereas if you put the energy of these separate pulses into a single pulse and excite it with 1 pulse, you have more fluorescence light. And that means that you can either reduce the input pulse energy or have higher signal-to-noise ratio. The next is that the choice of the lateral and the axial PSF for a fixed power budget is determined by the trade-off of the sensitivity of the neuronal detection versus the number of neurons that are detected, that is the volume size, which is basically saying that for a given amount of fixed power, for a given amount of power that we have, is it more important for me to detect all the neurons within a given volume, or do I want to maximize the total number of neurons that I want to record from the brain? And then the spatial sampling should be done at the Nyquist limit with a voxel spacing that is comparable to the lateral size of the excitation. I think this is self-explanatory. And then most importantly here, that the excitation of adjacent voxels in the sample should be separated only by the fluorescence lifetime and not more. So if we focus on this part and what I told you just before, you might have paid attention to this, that here the axis was actually broken. So if I were to draw that to scale, even with this 4-fold temporal multiplexing, you see that 85% of the interpulse interval still remained fully unexploited. So even with this 4-fold multiplexing, we are still waiting just for the next burst of pulses to arrive. So you can ask, so why not repeating the same temporal multiplexing over and over again? And the reason for that is simply practical limitations. So if you count the number of, or if you add up all the delay that you need to have in order to generate such a long delay lines for 30 pulses that would fit in here for the multiplexing, you end up with a delay line of more than 150 meters, which is not very practical in an optics lab. So to address this issue, we came up with a different idea. So we started off again with a high energetic pulse, and then built a cavity that we refer to as the maximum cavity. So we have a pulse that is coming in and is focused by a lens above the output coupler into this 4F cavity that has a slight imbalance. So the beam is focused here, it expands, and these are curved mirrors that are focusing the beam once again here, and then it focuses to this point. And then this mirror is slightly downward tilted, so tilting the pulse slightly downwards, such that it's hitting this output coupler again, where 8% of the pulse is coupled out towards the microscope, and the remaining amount of pulse is then again going a second round of focusing, and is then accumulating more divergence, and the process repeats again and again. So this cavity is solving now 3 problems all at the same time. So one is that the delay between for each round trip is exactly such that it is 8 nanoseconds. So it is exactly what you need in order to be beyond the fluorescence lifetime. The second is that since the first beam was focused, and by the time it reaches the output coupler, it has this divergence or this imbalance, each beam is accumulating divergence over each round trip of reverberation in the cavity. So subsequent beams are coming out at different angles from the output coupler. And the third is that this 8% input-couple ratio has been chosen to exactly match the loss of scatter-induced photons as a function of depth. So in total, we see then after reverberation of 30-fold, the pulse is starting off with a higher energy and goes gradually to lower and lower pulse energies. Each of these pulses are separated by each other temporally by 8 nanoseconds. And spatially, they are separated by 15 micrometer. And that distance has been chosen such that the divergences that we are getting here are in such a relationship to each other that's focusing them with a relay optics, and the microscope that comes afterwards causes them to be focused in different axial position in the samples. So we then coupled this output to a mesoscope, and which covers an area of 5 by 6 millimeter roughly. So when we scan down the beam, we are scanning these light beads along the axial direction within our sample. So we just look at the arrival time of these 30 foci and know exactly where which planes the signals were coming from. So using this approach, we can now record from a volume that is about 6.4 times 5 millimeter times 500 microns at something like 2 or 3 hertz. And here you see now the population activity of about 1 million neurons that we get in this way, that are distributed across both hemispheres of the mouse brain, and while the mouse is performing various tasks, etc. So here is then basically the heat map of these 1 million neurons, which is very difficult to make sense of. So you can start to zooming in. And even at 500 neurons, it's still problematic until you get to the single neuron level, where you can see we still get a reasonable signal-to-noise ratio for the individual neurons that we can collect. You can imagine that this is a large amount of data, and we are still in the process of making sense out of this, and both in our own lab, but also with collaborators. But one of the first things we can look at is, for instance, how these functional networks are being formed by these populations of neurons. And here you see what we found is that there are, for instance, 36,000 neurons that have correlation distances that are larger than 2 millimeter, providing support for the whole idea that I was mentioning before, that we have these long-distance correlation of neurons that are distributed within the brain. All right, so this made us also to step back a little bit and look at sort of what is the scaling of the simultaneously recorded number of neurons in various model systems in the lab, and what should we do next, right? So these points are showing the number of neurons that our lab has been able to record over the last 10, 12 years. And so if we look at them, and if we were to naively extrapolate from this, we have this Moore law, and so we would think that maybe in a few years we will be able to record from the whole brain. But there are...
Actually, you clearly haven't been reading the climate literature.
Yeah.
No, you would be at 10 to the 10 in 2027. You use the hockey stick model of data evolution.
Yeah. All right, so but we know that this is, the reality is way more challenging because of scattering, and depth, recording from neurons at depth is not such an easy feat. So we have to take these limitations seriously and then ask instead, what is it that we really need, right? So how valuable would it be to have a single cell resolution whole brain recording or a cortex-wide recording from a mouse? Not that it wouldn't be valuable, but how much quantitatively would it be valuable? How much more information is there as opposed to, say, half of that size of the recording or a third of that? And ultimately, if we have a limited amount of resources, how should we best think about the most efficient allocation? Right, so this brings us a little bit back to the same problem that I started with. So we started to ask, now that we are no longer here, where we are recording from only 100 neurons, can we say something more about maybe the amount of information that is in these neuronal recordings, and that may guide us to further or better allocate where we should be putting our resources for acquisition? So one point that I want to highlight, which is a little bit ironic, is that if you look at many publications that are recording from hundreds or thousands of neurons, you first record from these large number of neurons, and then the next thing you see that most people start doing dimensionality reduction. So you get rid of a lot of the information that is in there. So this is for an example from our own publication in 2013, where we did whole brain recording in C. elegans, and then we found that 75% of the variance is included only in the first three principal components. So the question is that what about all the other components? Why is there that then in the brain? So I think one of the key caveats of these dimensionality methods is that it's very difficult then to say whether these components are containing noise or signal, because there is always sort of an ad hoc choice made about up to what level we want to consider, and the rest is typically just ignored. So to address this question, we adopted another technique, shared variance component analysis, that was developed by Carson Stringer and in Matteo Carandini's and Ken Harris's lab. So in this approach, we are splitting neurons into two sets, and then we are looking at the joint embedding of them in this lower dimensional space, and then we test then the lower dimensional dynamics that we are inferring on some withheld timed data points to establish the reliability of this dimensionality reduction. So in a way, we have an objective way of knowing up to what level of information that we are containing here is signal versus noise. And so when we apply SVCA to this neuronal population recording, what we saw is that there is a very smooth decay of this variance spectrum up to the level of, here we find to thousands of SVCs, and there is no obvious point where there is a significant drop. So it's not clear whether there are any of these or when they would be encoding noise. So here you see now an example of that for, I believe this is 120,000 neurons, where you can, and this is the comparison to the shuffled data, which is establishing a noise floor for us, and clearly up to maybe 1,000 of these dimensions remain highly reliable. And not only that, but also if we look at this as a function of number of neurons that we record, we find that as you are going to larger and larger neuron numbers, the dimensionality that we find in these dynamics seems to go up as compared to what we have here as the objective noise floor. So that may be explaining why maybe if you can record from, say, 100 neurons or 500 neurons, you may find that the dimensionality that you have is on the order of maybe 10 or so. But then this by itself shows that the information remains still unsaturated, and this is shown here, even up to 1 million neurons. So as we are recording past 1 million neuron, we are still adding significant variance to our data that is distinct and different from noise. And this seems to be conserved across different animals. Now, all of this is obviously opens up a number of questions. So what are these dimensions and encoding? What portion of these what portion of these are encoding for spontaneous behavior? And what are these SVCs? How are they anatomically and temporally organized? So these are some of the things we would want to follow up on in future studies. So with that, I just want to thank the people who did the work. They are marked here, who contributed, as well as some of the former lab members. I'd like to thank our funding sources. And we have also positions if any of you should be interested. Thank you.
Thank you so much, Dr. Vaziri. I think we have time for one question.
So you said that you have this grid which you completely sample. It's not sparsely sampled, right? Like you measure every single voxel. How do you do the neuron detection? Because most of the grid will be empty, right? And at the end you show these nice blobs. Like how do you do that detection? And are those blobs reconstructed? Like is it just a 3D model, or is that just what your data looks like? Because that's how good your
Yeah, so this was just a representation or a rendering of what these individual I shouldn't say spots, but neurons are. So this, the spacing is, depending on the size that we are recording, is somewhere between, say, 1 to 5 microns. So we have, on average, something between 9 to 25 spots on each cell body that we record from. So, and then we can, sending then the data to data analysis pipelines such as Cayman or Sweet2P, we can segment these cells, and this is what you were seeing as blobs there.
My apologies, quick follow-up. Have you ever used that in order to do a sparse sampling afterwards, now that we know where the neurons are? We don't need to sample.
Yeah, no, we haven't done that yet. But I think that's a great way of limiting the exposure of power to the sample and things like that, yeah.